All right. Good evening, everyone. Thank you all for joining us today. It is my great pleasure to introduce our esteemed speaker, Brendan Swain. Brendan's journey in science began right here at the University of Cambridge, where he pursued his studies in natural sciences. His academic path led him to delve deeper into the field, where he undertook a PhD in the pharmacology department. It was during this pivotal period that Brendan, alongside his colleague Daniel Ives, laid the foundations for what would become Shift Biosciences. And Brendan will talk to us today about unlocking safe reprogramming with deep learning. So, Brendan, over to you.
All right, hopefully that works. Yeah, so with that introduction out of the way, I just want to briefly talk about the structure of the whole talk, and then I'm just going to derail it for a moment. But largely I'm going to talk about some of the science background, because I'm not sure what the balance of longevity society people to science society people is, so I think there's some background that non-field specific people will need. Then I'm going to talk about one of Shift's major breakthroughs, which is our single-cell age predictors. And finally, I'm going to talk about the problem of combinatorial space and why it is a problem in our field and how Shift is trying to deal with it. But I'm just going to talk about my career progression so far. So, Brandon did talk briefly about it, and I'll also talk briefly. And it's not a careers talk, so I'm not going to go into CV level detail. But I did my undergrad here in natural sciences, did my PhD down the road, and then I went straight to Shift to take on an executive role. So it's quite a strange journey, and it's been quite a short career so far. I'm not going to go into too much depth about it, but I think there is some good learnings there, especially if people are doing PhDs. I did an internship as part of my PhD, and then that's basically why I'm here today. So the lessons from this journey so far have just been take opportunities and take advantage of the training that Cambridge offers, because there's a whole host of free stuff. So, bioinformatics, machine learning, all those kind of Python-y courses. I definitely recommend getting involved. And if you're interested in talking more about this slide, you can come grab me afterwards or ask a question at the end. So just back to the main agenda, talking about the background of the science. So for most of you, I'm sure the question, why would you want to tackle aging, is pretty obvious, but just if the answer to that isn't immediate, I've got this slide which I stole from Yuri Dagan, actually, at Life Biosciences. So there's a statement that we could probably all agree on, which is that aging causes a general loss of resilience and increased frailty. And, most of us in this room probably aren't actually really going through that yet, but we'll definitely know people who are. And to kind of enumerate the problems that come along with aging, basically all the organs and body systems start to fail. And the rates at which those organs and systems fail differs depending on which organ or system it is, but they basically all fail. And therefore, if aging causes these problems, it seems pretty logical that if you then target aging, maybe you can prevent them, maybe reverse them. And I think a useful thought experiment, it's not an original thought of mine, but if you think about us discovering a disease tomorrow that would eventually affect 100% of the population, that led to a degeneration in all these body systems until the person dies because of those diseases, you'd be pretty motivated to find a cure. It would be a COVID-level response to that. So I think, that is sometimes a surprising thought to people, but something that I should hammer home. And we've seen an explosion of funding and research going into this in the last 3 years, basically. It's just become a legitimate field, basically, according to most people. So I'm just going to place Shift in that broader landscape. And Shift is based on two major discoveries that I'm sure some of you are familiar with, and the first relates to measuring age. So if we are trying to reverse age, prevent it, we need to be able to measure it in the first place to know when we've struck gold. And if we were doing an experiment with aging, let's say 20 years ago, we could very easily start that experiment. We could take cells from a donor, and we'd know their chronological age because we could just measure how long it had been since that person was born, or how long they said it was. We could do our intervention. We put the cells in a dish, add a drug, or whatever the intervention is. But then at that point, we'd kind of be a bit stuck. We could perform assays that are related to aging, but we couldn't necessarily say for sure that we'd tampered with the core biology of aging. And that changed in about 2013 or so, when it was discovered that you can actually predict quite well the age of a cell sample by looking at the patterns of methylation on the DNA in those cells in that sample. And Steve Horvath discovered that actually you can build a single one of these clocks, as they're called, for all the tissues in the human body. So there's something conserved going on with the biology of aging in humans. And I think it was last year, actually, it was discovered that actually, no, it's not just humans. You can build a single clock that ticks in every single species and in all the tissues in all of those species. So there's some really conserved aging biology that's happening across organisms, which means you're really tapping into something at the core of the process. So that's all well and good. Chronological age is cool, but ultimately, if you know a person who's 70 years old, you can predict generalities about their health, but you can't say for sure exactly what their health is going to look like. So this motivated the development of more phenotypic measures of aging. So you can build these clocks and actually have them relate more to what's now called biological age. These more phenotypical measures, such as, does the person smoke? What does their blood biomarker profile look like? And kind of morbidly, how long is it going to be until that person is likely to die of natural causes? So as well as chronological age, we can now link essentially DNA methylation to clinical outcomes and clinical phenotypes. So when we find something that prevents or reverses age, we're pretty confident that we're going to see it. So that's the first discovery. And then the second is actually based on that. It's finding an example of age reversal. So you're almost certainly familiar with the Yamanaka factors. They won the Nobel Prize in 2012. They take an adult stem cell, you overexpress 4 transcription factors, and an adult cell, you express 4 transcription factors, and you make a stem cell. It won the Nobel Prize. It's transformed bio research. It's leading to therapies in the clinic. But a couple of years ago, it was also shown that actually it reverses the age of those cells according to these DNA methylation clocks all the way to 0. So people were understandably very excited by this. If you can reverse aging, maybe you can reverse all of those downward lines that I showed you before, make people healthier. And indeed, people have then gone on and used the Yamanaka factors to achieve this kind of thing. It's very difficult to run clinical trials, especially in humans, that extend lifespan because the FDA doesn't recognize aging or dying of old age or disease, but you can focus on diseases that are often age-related. So there's some in vitro data where you can take human chondrocytes from osteoarthritis joints. So that's a bone disorder that you get, or older people tend to get. You can reverse that phenotype. And then in vivo as well, you can target the eye, and in mice, reverse glaucoma, and in non-human primates, target a very rare eye disease and seem to ameliorate that as well. So there's good evidence that the rejuvenation that comes with OSKM is linked to disease reversal as well. But these factors were discovered because they make cells into stem cells. So along with this reduction in age, you also get an increase in pluripotency, as you should expect. But pluripotency in a clinical setting is not a good thing. So this is a grotesque picture that I'll blur, but where these circles are, there were massive tumours, which are multi-tissue tumours called teratomas. If you induce pluripotency in an animal or a person, you make tumours, which is not a good thing, I'm sure you agree. And if it happens in a clinical trial, it probably ends the clinical trial and probably ends the company that ran the clinical trial. So we're not very interested in that, and we'd like to avoid it as much as possible. And that's what we're trying to do at Shift. So in this quite simple graph, we're trying to move cells from the right to the left. We're trying to reduce the cell age, but not change the cell identity. We want them to stay low on this y-axis. And as I said before, we have this evidence that you can do this kind of diagonal movement in this graph. You can change the cell identity, change the cell into a stem cell, but you also reduce its age, so that's promising. And what's been shown more recently is that if you turn on the Yamanaka factors for a very specific length of time, cells begin to progress towards this diagonal line. They lose a bit of age, but they also start to lose their cell identity. And then if you turn off the expression of the Yamanaka factors, let me just turn a pointer on here. If you turn off the expression of the Yamanaka factors at a day 13 time point in this paper where it was shown, the cells kind of bounce back from their progression towards pluripotency, but they keep this reduction in cell age, meaning that you can somehow separate these two phenomena. So that's very promising for us. And then finally, the final kind of evidence is that, but we at Shift, we don't really believe that this is the right approach to kind of make Yamanaka factors or reprogramming more generally safe, because it's very easy to control accurately the expression of cells in a dish, but it's quite difficult to do it in a human or in an organ or any other organism, because this needs to be a very controlled process. If you leave it for too short a time, you don't get rejuvenation that persists, and if you leave it for too long, again, you risk pluripotency. It seems to be a time window of 1 or 2 days in the dish, which is nearly impossible to control gene expression to that resolution for time and cells in an organ or an organism. And even if you could do that, it's feasible that we could do that in the future, maybe. But given that you actually have this progression away from the original cell type for a period of, a week or 2 weeks, that's actually quite problematic if you're doing it on an organ level or an organism level. You have function for a reason, and if you're losing function for a prolonged period of time in a lot of cells, that's bad. So that's why we're not focusing on this angle at Shift. And then just a final piece of evidence is that you can actually change cell identity without changing cell age. You can transdifferentiate cell types directly into other cell types, and they are old cells that come out the other end. They don't actually de-age. So you can decouple these two axes that I've drawn up here. And therefore, given that we can decouple the two axes, we want to basically recreate this movement on the x-axis. We want to de-age cells without having any concordant change in cell identity. And just to kind of frame this question a little bit better, what we're looking for is a combination of transcription factors that can do what the Yamanaka factors do, but safely. So if we assume that we're looking for 3 transcription factors, given that there are 1,300 transcription factors, give or take, that means that we're looking in a space of 365 million combinations of transcription factors, which is large. And so I've represented that with this grey box. This is all of those combinations essentially smushed together. And we are trying to map this landscape, find the areas of low rejuvenation, find the areas of higher rejuvenation, and therefore understand the peaks of safe reprogramming in this landscape, which represent combinations or groups of combinations that can safely reverse the age. And then what we are going to do that is using aging clocks. As I mentioned at the beginning, that's one of Shift's big breakthroughs, using cell simulations, map that entire landscape, find all those peaks, of which there are likely several, and then now that we have a list of all these interventions that safely rejuvenate, we can just pick and choose which ones we want to use for our preclinical or clinical programs. So that's, in a nutshell, what the program is. And to be able to do this, there's 2 capabilities that we need to have to even have a shot. So the first is measure aging accurately and rapidly. So, once we find a rejuvenative intervention, we need to know that we found it. We also need to be able to move through that space quickly. So these wet lab tests need to be very efficient. We need to have good clocks, essentially. And not only that, because the space is so big, we can't hope to search all of it in the lab. So we need to be very choosy about which experiments we're doing in the lab. So that's the second point. We need to explore that combinatorial space very efficiently. So going to focus on that first point, single-cell age predictors, measuring age accurately and efficiently. And what do we need for a single-cell age predictor? Well, basically, the only things we need from a predictor are we need to have confidence in it, and we need to have confidence in it by measuring our intervention in as few cells as possible. And this is just for practical reasons. If you have to measure tens of thousands of cells with an intervention to know if that intervention rejuvenated, it's going to take a lot longer than if you just have to measure an intervention in 100 cells to understand what the intervention did. So that's the ultimate aim of making an age predictor in our program. And if you think about this problem, what makes it easier to be confident about the effect of an intervention? Three main factors kind of rise to the top. So you have effect size. It's pretty obvious. If you have a bigger change, it's easier to see. So a cell that is de-aging a lot is easier to detect than one that is only de-aging slightly. Intervention efficiency. So this will become clear why I put this later, but if a rejuvenation intervention only rejuvenates 1% of the cells that it gets into, it's going to be a lot harder to detect than one that just homogeneously rejuvenates the entire population. And then finally, aging clock accuracy. So you'll notice that the first two we have no control over. That's just biology, whether that intervention exists and whether it's good. But we can train a better clock. That's one thing that we do control. And so at Shift, we have a very specific approach. And I've talked about the epigenetic clock so far, and that is the gold standard of age prediction so far for the field for very good reasons. One of the main reasons being that there's very little batch effect in DNA methylation data. So if I take a cell sample, measure the pattern of methylation on the DNA in that sample, and then send those cells to someone else across the world, it's quite likely that those measurements are going to look quite similar. And therefore, massive public data sets have been built where data from different sources has all been chucked together. It's all pretty compatible without much work. It's pretty much out of the box. And so this has just been a playground for smart people, essentially. Lots of data, and they've had a lot of time as well, because that data has existed for over 10 years at this point. So we have these very accurate and well-validated clocks because there's just a lot of time, a lot of data for these people to work with and build good predictors. And not only that, because there's a lot of metadata attached to those data sets, they can also build predictors of other things like those phenotypic clocks I talked about and other things. However, DNA methylation is a bit limited for age prediction for a couple of reasons. So the first is that currently, age prediction using methylation requires you to take a dish of cells, scrape them into a tube, and then essentially process that for methylation, which means you are measuring the average methylation level at each site in the DNA. And so you can only get a population level average for the methylation age when you run it through those algorithms. And that means that your screens become very slow and quite expensive because every intervention needs its own dish or its own well. And so processing it from a practical perspective is just a bit annoying. So we wanted to deal with that and essentially introduce single-cell transcriptomics into the equation. So just to kind of compare it and contrast it with DNA methylation, there are actually quite large batch effects in RNA-seq data. So, if I measure the transcriptome of some cells, it's likely to look quite different to someone who does that on the other side of the world. So these massive data sets of kind of compatible RNA-seq data from single cells don't really exist. I mean, they're starting to exist now, but it's been a long time that it's just been kind of inaccessible as a resource. And so we don't have these really high accuracy public domain clocks. We don't have very well-established phenotypic clocks. But what we do have is a really standardized, easily accessible way to measure single-cell transcriptomics. And therefore, if we are running screens with an age predictor based on single-cell transcriptomics, we can make it much faster and very parallelized, because suddenly a dish can have many interventions in it because you'll just measure the transcriptome later and work out what those interventions were. So what we've done at Shift is to make our aging clocks, so AC just for short, and we've tried to capture the best of both worlds, the good things about single cell, the good things about methylation, and try and combine them into one thing. So to do that, we built, as far as we can tell, the largest data set for aging fibroblasts that exists. We took cells from over 100 donors of different ages, took fibroblasts, skin fibroblasts, and the first thing that we did was DNA methylation analysis to work out what is the biological age of these donors. because chronological age is not a very good label for several reasons, and so we wanted the best ground truth possible. We then also collected single-cell RNA sequencing data from individual cells from those same samples. So now we have paired measurements of single-cell RNA sequencing, and we have labels for what age those cells are according to epigenetic clocks. And then what we did is we used one to predict the other. So given that we know how old every cell was according to biological age measurements, we can use the gene expression vectors for every cell to make that prediction. And that is the basis of our clock. We use those gene expression vectors, and then we can take advantage of all of those years of development, all of those massive data sets that we used to build the epigenetic clocks, and leverage that in our single-cell transcriptomic clock. And so the first advantage of that is very obvious. It's highly parallelized, as I said before, but a couple of more subtle points is, because you're measuring it in every single cell in the dish, if, for example, an intervention is not 100% efficient, as I mentioned before, like OSKM, you actually still would be able to detect it in your initial screens. So OSKM actually only reprograms about 1 or 2% of the cells in the dish. So if you just bulk methylome epigenetic clock analysis OSKM cells, you actually probably wouldn't detect it until very late in the process. So, given that the current gold standard intervention for rejuvenation isn't detectable by DNA methylation clock screens, it probably tells you that it's not very good. Those clocks are not very good for that purpose. And then finally, just because you're measuring the single cell transcriptome, you know what each cell looks like. It's very easy to measure markers of pluripotency, cancer, things like that, just to make sure that what you're doing is actually safe. So these clocks are pretty great, and just to give you an example of the performance of these clocks, so the AC2, aging clock 2, was an early iteration of these clocks, and this is just the performance of that clock. On the x-axis is the true biological age of the sample, so the epigenetic age of the sample. On the y-axis is AC2, the prediction for single cells in that sample, and that's why there's a distribution of ages around the mean. And you can see that the prediction is pretty good, that the mean predictions are pretty close to this y equals x line, this dotted black line. And to put a number on how good the clock is, we can use a Pearson coefficient, which is a maximum of 1, where you have a perfect clock where all of the predictions lie perfectly on the y equals x line, down to 0, where the clock is completely useless and it gives you no information about aging at all. And this clock has a Pearson value of 0.8. So it's pretty good, and it's actually better than we thought might be possible with this data set. So we're pretty happy with that. And then now we're actually at AC3, so we've gone we've added 1 to the number. There was a non-obvious ML trick that made the clock better. And the other change that has happened in this graph is that we've added more data to this independent test data set. And that gives us this final estimate for the performance of this clock of 0.8, 0.9 or so Pearson correlation coefficient. So we have a really good clock. But, at some point, just improving this number doesn't really mean anything. It's kind of hard to link this to the real world. So we try to do that, essentially. We try to go from Pearson correlation coefficients and convert that into something more meaningful, like what is the cost of a program given a clock that is this good? What is the speed at which you can search combinatorial space? So we ran some simulations. I won't go into the detail, but essentially what we found is that as you progress from one of our earliest clocks with a Pearson correlation coefficient of 0.7, and that's where we imagine most people who embark on making one of these clocks will actually end up. As you progress to 0.9, you actually get a 4 times reduction in the amount of money that you have to spend on every test, and therefore reduce the cost of your program 4 times. And you also have a concordant increase in speed. And to be honest, speed is actually more important because it's a very well-funded field at the moment. You've probably heard of Altos, and there's several other very well-funded competitors. So money isn't so much of an issue in this field. It's really about speed. And so that's what we tend to focus on. So our AC clocks are pretty great, and to be honest, they're almost as good as you can get. There's not very much more room for improvement. So we tackled that one problem, but the second is even greater. So we've got to explore combinatorial space effectively. And just to go back to this diagram, we want to map this landscape, find the contours, find the peaks. So, one kind of naive approach would be to say, well, let's just test every combination. Just do single-cell RNA sequencing with all these interventions, all these combinations of 3 transcription factors, and just see what happens to AC3, see what happens to the age prediction. Find the most rejuvenative, there you go, that's your drug. Unfortunately, this is a very large space, and so in this tiny black dot, this is a 2-scale representation of the number of interventions that you could test with a lot of money in a 1 or 2 year period. So you can see that you're talking in the order of tens of thousands of combinations that you can test in a normal lab cycle amount of time out of 365 million. So it's a very small black dot. It's a very big grey square. So we really need a way to navigate efficiently, because we can't just rely on accidentally hitting the right peak with that tiny black dot. We need to make sure we spread it out in a very efficient way to maximize the probability of seeing a peak. So how do we do that? Well, we have 2 main tracks. The first is relatively simple. I won't dwell on it too long. We have a lot of OSKM data in-house. We've generated kind of proprietary data sets using different data modalities to work out what genes are driving the OSKM rejuvenation events that aren't linked to pluripotency. How can we extract the rejuvenative biology from that process without driving pluripotency? And that defines a very small space in combinatorial space that you actually can search exhaustively. So this is actually not to scale representation of the space. It's very small, and we are testing that in the wet lab. But what we're more excited about is, because although you can discover the first safe reprogramming combinations using this left-hand method, you can't discover them all. And I'm sure we're all familiar with the risk of getting scooped. It's something that most people worry about. If you alight on a single peak and you take that to clinical development, there's always the possibility that someone comes along, they found a better peak just by chance, perhaps, and suddenly your whole company is worthless. So what we're more excited about is track 2, which is basically map the entire landscape, find all of those peaks of safe reprogramming, and then you've won. You have this long list of those combinations, and depending on the different features of each combination, you can choose one for a certain disease, one for another, and you're free to do as you wish. So we're very excited about track 2. And the way that we are achieving track 2 is using what's called active machine learning and cell simulation. So what is active machine learning, first of all? Well, active machine learning is basically just a fancy way of saying iterative wet lab testing. So we use models to predict combinations of genes that are rejuvenative. We test those in the wet lab. We collect single-cell transcriptomes. Because we have AC3, we can therefore say what age every cell in that dish is. And we also know what transcription factors each cell in the dish got. So we can retrain our rejuvenation model, our active machine learning model, by saying, yep, you were right about this intervention, it rejuvenated the cells, but actually this was a rejuvenative and you didn't expect it to be. The model incorporates that information and then lets us predict new gene combinations that we should test next time. And so we just run this loop until we find something interesting, and we kind of spin out into this preclinical area over here. So effectively what active ML does is it identifies areas of this landscape that we need to test in order to understand the entire landscape. And what I've represented that with here is these blue squares. So we've basically identified these small regions that we need to test to understand the entire landscape. And we estimate that this active ML approach will actually reduce the space that we need to test in the wet lab to about 3% of total space. So, we're getting a really good reduction here. It's like 30 times. It takes it down to about 9 million combinations that need to be tested. But what you'll remember, and then we just map the landscape. And unfortunately, what you remember is that this black dot that I showed you before is very small. So the number of interventions that we can test is still much, much smaller than an active machine learning program, which is basically the height of drug discovery at the moment, can discover. So we still need some way to make sure that we can test all of these blue squares to fully map the landscape. And that's where this concept of cell simulation comes in. And it's quite a small addition to this loop. Essentially what we can do is, rather than adding transcription factor combinations to cells in the dish, we can just do it in the computer. We just tell a computer model, I'm adding this transcription factor combination to cells. What does that do to gene expression in single cells? Again, we have AC3, so we can then feed that gene expression into AC3 and say how old cells would be given a certain transcription factor combination, and therefore what is the most rejuvenative transcription factor combination. And what that lets us do, therefore, is start filling in these blue dots. So start converting the blue dots, i.e. identified areas, into black dots, tested areas, and just kind of progressively go through. And because in silico simulation is much cheaper and much faster, more importantly faster than the wet lab, you can actually do this task. You can fill in those regions that you need to test very quickly and therefore map the entire landscape. So how do we do that? Like all machine learning, you need to train a model, you need to give it data. Luckily, we have some good data for that. We have a screen of 733 transcription factors where we individually overexpressed those transcription factors and took single-cell RNA sequencing data. We can then use that data to train models. So a good example is a public model called GEARS out of the Stanford lab. It currently seems to be the gold standard public model for these in silico perturbation experiments. So what are you feeding that model? You basically say, well, I've perturbed or I've overexpressed this gene. This is what the gene expression vector from this cell looked like. You train it on that. And then what we can do is use that to predict the effect of expressing combinations of those transcription factors. Then again, because the output of the model is some gene expression vectors, we can just use AC3 to say which is the most rejuvenative combination of transcription factors. We can then test those in the dish and then feed that data back into this model, iteratively increasing the accuracy of the cell simulation and therefore making us more able to shift experiments into the computer versus doing them in the wet lab. So again, we try to do this kind of thought exercise of essentially making that tangible. What happens as you make a better simulation? What effect does that have on the speed and the cost of your program? So to start with, if you just consider this column here, this is where we currently are. We have AC3, our very, very good wet lab clock, where we have to perform 100% of tests in the wet lab. To do this kind of whole program, it would cost us about $3 billion, which is a lot, but it's not insurmountable. But importantly, the time it would take to do a program based on a wet lab only testing system would be hundreds of years. So it's basically completely infeasible for anyone to ever test this problem in a meaningful way in only the wet lab. But as we move tests from the wet lab into the dry lab, we basically massively reduce the cost of the program because we're doing much more of it in the computer. And again, importantly, we also really increase the speed. So it goes from hundreds of years into the order of single-digit years, which is very important. And as I said, it's the most important factor in this field, both in terms of making things practical and competitively. So by developing these simulations, you can basically actually test this limited subset of the landscape that you need to fully understand it, and then find those peaks and develop a drug. So just to finish, this is the team. So we have Daniel, the CEO, me, Lucas, who's actually sitting right here, he's the head of ML, and we've got a great wet lab team as well, Kate, Andreas, Raya, and Samira, the wet lab team, responsible for all this work. And we've also got Steve, the CFO, Charlotte, our executive assistant, and we've got some great advisors as well. So I will leave it there. Yeah. Okay, yeah, any questions? Oh, I'll hand over to the compere.
Hello. Okay, do we have any questions for Brendan?
Thanks for an amazing talk, very inspiring. So if you're allowed to answer, I would like to ask what kind of cell type you use, how you express the transcription factors and for how long, and why also did you choose 3 instead of any other number of combinations?
Yeah, so we use dermal fibroblasts just because it's easy to get your hands on dermal fibroblasts. Cosmetic procedures and relatively low invasive procedures you can get dermal fibroblasts from. Not going to talk about gene delivery. And then the last question was why combinations of 3, and it's a good question. So on a practical level, combinations of 4, no one can really ever hope to search that space effectively. So that's one reason. Then the other reason is actually that the current gold standard for in vivo and in vitro rejuvenation is OSK, so 3 out of the 4 Yamanaka factors. In non-human primates, that's enough to rejuvenate the eye. So it seems likely that 3 genes is probably going to be necessary to do something similar in a safe way. And that's pretty much the rationale for it, yeah.
And how long do you express them for?
Yeah, I'm not going to talk about that either.
At this point, you only use the single-cell RNA analysis to determine age reversal, or do you also do methylation still in AC3?
Yeah, exactly. So AC3 specifically is only RNA, but it's essentially like the classic high throughput screen, low throughput validation afterwards. So AC3 is our high throughput measure, which identifies the hits, and then anything that looks promising according to AC3 goes for DNA methylation.
Thanks for the talk. How well do the individual, speaking is difficult. How well do the factors do by themselves in terms of reducing age? Do any of them have any significant impact? Or is it only with the impact of all 3 you see something significant?
Yeah, so it depends on cell type largely. So in some cell types you only need O and S, but it largely seems to be a kind of threshold-ish effect where you need at least a combination of 2, normally 3, to have this large rejuvenation effect. But there is some correlation of just single genes with rejuvenation, but we're not sure yet whether that actually is meaningful and irreversible.
Thank you for the talk. That was very interesting. I'm not familiar with the field, but how successful and commonly used are the cell simulation models?
Sorry, could you say that a bit louder, please?
How commonly used are the cell simulation models, and how good are they?
How commonly? Oh, so they're hot off the press, basically. So they're pretty novel. I think. So you might have heard of SCGPT. So this is basically, riding off the back of ChatGPT with the name. It's a transformer model that does exactly this job, and that only came out this summer. So it's quite a nascent field, but it's surprisingly good. The models are surprisingly good, and not only that, actually, even with a kind of what seems like a not very good model, it's only accurate a third of the time, let's say, you actually get a surprisingly high impact on the amount of wet lab work that you need to do. Even if you can't fully trust the simulation, it still really drives down the number of wet lab experiments that you need to do.
So my question is regarding the cost of the simulation. So what is the allocation of this cost, and are there any opportunities to reduce it, potentially by a lot, potentially simulating all combinations?
Yeah, it's a good question. So it's not something we've really explored in huge depth yet, just because the advantage of going to the dry lab in the first place is so massive compared to the wet lab. So it's kind of diminishing returns at the moment. But these models currently are, depending on which one you use, are quite large, so they are quite computationally expensive. So there's a lot of scope for reducing their costs. Yeah.
So thank you for the talk. I have 2 kind of broad questions. The first one would be, so we have these combinations of transcription factors that can reverse the cell fate. How do we apply this in a more clinical setting in the future? And then the second question would be, okay, I forgot. Sorry.
Makes my life easier. Yeah, so it's a good question. I think we're quite lucky in the current era where suddenly genes are actually targets, and they're also drugs at the same time. So you can deliver virus or mRNA and actually have it express in the right area. So if I go to those in vivo studies that I mentioned, in the non-human primates, it was an injection into the eye, and the eye is actually a very promising location for these kind of programs because, and for the same reasons it is for other gene therapy, because it's shut off from the rest of the body and it's very contained, and you can basically hit all the cell types because you just inject right there. So they used AAV2, I think it was, and basically overexpressed these factors. So we envisage something similar, just because it's kind of easy as a proof of concept in vivo. But eventually, people are working on adeno-associated viruses, AAVs that can get to the whole body or to specific organs. So it seems pretty feasible that you could have a whole body treatment with these kind of therapies in the next 10 years or so.
Yes, and the second question would be, so a lot of aging-related diseases, they usually involve a lot of accumulation of mutations across a individual's lifespan. So does using transcription factors to reverse cell fate kind of reverse these mutations, or would that be a concern of those being accumulated more and more?
Yeah, so I think there's kind of two elements to that question. So the first is, if you had a preventative treatment, you could reduce the rate at which those mutations accrue, obviously, but also as you say, rejuvenating, i.e. reversing the age, isn't necessarily going to change the mutations that are present in that DNA, but actually most cells are given rise to by stem cells. So if you can rejuvenate stem cells, you can replenish the healthy population of cells in the tissue at the expense of those unhealthy ones. So I don't think mutation is really going to be too much of an issue, actually. Yeah.
Once you find a sequence of transcription factors that rejuvenates the fibroblasts effectively, would you then test other tissue types, or would you just keep that as the general formula?
No, exactly. You're exactly right. So there's kind of two angles for this. One is clinical development, and the other is just generality, because I think the field generally expects there to be a combination of genes that safely reprograms everything, rejuvenates every cell type, and that might be a bit much to hope for. But the first, we find the combination. The first thing we do is we test in cells from the 3 different germ layers, because that's a good argument for generalizability if it rejuvenates all 3 germ layers. And then the second is go to clinical cell types. So, for example, eye cells, retinal ganglion cells. If you can rejuvenate those, it seems pretty likely you'll affect glaucoma and nigh on that rare eye disease. So that's the immediate thing. But yeah, ultimately, fibroblasts aren't enough, but they're just the vehicle by which we discover this biology to start with.
On the topic of tissue types, do you think there are any specific cell types that might be resistant to reprogramming, or do you think, what's your opinion on that?
Yes, good question. I think one that I was worried for a while that we wouldn't be able to target very well was neurons, but actually, and the reason being, there doesn't tend to be much cell division in neurons. So if a cell is irreversibly old, maybe actually you can't really do anything about it. But actually, these non-human primate models showed that you can rejuvenate neurons in the eye, and that leads to a functional benefit without any cell division. So, to be honest, at this point, I'm pretty bullish on all the cell types. Yeah, I'm sure I'm probably wrong, but most, if not all.
Cool. Does anyone else have any last questions? All right. If not, can we give one more round of applause for Brendan Swain?